So good morning, everybody, and welcome. My name is Mark Whelan. I'll be your host today. I'm also the program manager for the Technology Gateway program, who are hosting this series of webinars. So the idea of the series of webinars is really just to provide insights into new technologies and interesting areas that we hope for industry. So the plan is to run a number of webinars over the next couple of months, starting today, and then into January and all the way to June, on just different aspects of business and what hopefully people will find interesting. Sharon, could you just forward on the slide for me there, please?
Sure.
So as I said, the series of webinars has been hosted by the Technology Gateways, which is the program I run in Enterprise Ireland. And the idea of the gateways is that they're an avenue for industry to engage with the Institutes of Technology to find solutions for problems that you may have. So what we really wanted to do was kind of share some of the insights that we have found over the years and some of the new technologies that are available to industry. So Sharon from GMIT, I'm delighted to welcome here today, is going to present the first case study first, pushing the boundaries of in vitro testing. So the way we will run the program today is Sharon's going to do the presentation. And you're able to ask questions. If you see on the clipboard that you have on the side there should be a tab for people to ask questions that we'll collate and keep to the end. So the idea is that when we get to the end of Sharon's presentation, we'll spend about 10 or 15 minutes doing questions and answers. So if you have them, feel free to put them in at any time. But as I said, we'll address them at the end. So I'm going to hand over to Sharon now in a moment. I'll come back at the end. And I hope everybody enjoys the webinar. Sharon, over to you.
That's great. Thanks very much, Mark. I'm just going to switch off my camera here. Hello, everybody, and many thanks again for taking the time to join us for our webinar today. As Mark mentioned, my name is Sharon White, and I'm the Technology Gateway Manager at the MedTechnology Gateway. I have over 13 years experience in the medical device industry with a background in R&D and regulatory affairs. I joined the Met team 3 years ago, initially as a senior research engineer, before taking up the role as the manager in 2019. And I'm delighted to have the opportunity to showcase some of our capabilities in vitro testing to you today. For those of you who don't know us, the Enterprise Ireland Medical and Engineering Technology Gateway is an interdisciplinary applied research centre dedicated to supporting research, development and innovation activities in the MedTech, life sciences and engineering sectors. Through the Gateway, we provide open access for industry to the academic expertise within GMIT, and we generate solutions for the close-to-market needs of companies. MET in its current form was established in 2016, but the research and technologies behind MET's current offering were developed over the 10 years before its establishment, and our researchers continue to build and expand our capabilities. We have a team of over 20 researchers from a range of disciplines, including biomedical engineering, physics, biomedical science, data science, and medicinal nutrition. Through active engagement and collaboration with our clinical, industrial, and academic partners, we produce scientific knowledge and technology outputs across a range of streams. We offer support to industry across 5 technology themes, the first 2 of which will largely constitute the subject of our webinar today, and you will hear more about them a little later on. Our third technology theme is data analytics and visualization, where we have expertise in data analytics and the visualization of clinical data to inform the design of next generation medical device prototypes. Our fourth technology theme is design engineering and verification, where we offer services in prototyping and design verification technologies, such as CAD design, rapid prototyping, technical reviews, critique, and materials and product characterization. And last but not least is our medicinal and nutrition sports technologies, where our research team with expertise in nutrition and sports science assists the food industry in all aspects of novel food development, including food analysis and testing, human intervention trials, product innovation, development and optimization. Additionally, the range of sport testing equipment and technologies available at the centre enables companies to develop nutrition and fitness strategies specific to athletes and those involved in sport. Could you do the next slide, please? Okay. Met's expertise and understanding in the in vitro modelling is based on our long history of biomedical research. This research has centred around understanding the disease condition and how it is influenced by physiological parameters and patient-specific anatomy. A large portion of our research is in the vascular space, in particular the central vascular system and neurovasculature, and has included areas such as abdominal aortic aneurysm, complex aortic arch aneurysms, and stroke. One of our most recent publications in the area of stroke, and published in the medical journal of the same name, evaluated the trajectory of clots through a patient-specific model. It analysed the effective parameters such as clot type, flow conditions such as atrial fibrillation, and the number of clots released on the likelihood of stroke occurrence. In our research on the treatment modalities in complex aortic arch aneurysms, we have evaluated the effect of endovascular intervention on disease progression while assessing the device performance, and we will discuss this in a little more detail later on. One of our most recent research studies was on congenital heart disease, where our models were used to predict the clinical outcomes of different surgical approaches. Our ability to accurately model the clinical condition is what underpins the value of our research and creates the basis for our future work. Next slide, please, Mark. From this research, we have developed unique expertise in the design and fabrication of bespoke anatomical models. Based on clinical data, our models may be designed to represent an idealized model of the target anatomy or be directly reflective of the clinical condition. As you will see a little further on, we specialize in the fabrication of patient-specific models, and in particular, non-planar complex anatomies. Our vascular models are fully compliant and directly replicate the vascular response to pulsatile flow. In order to accurately replicate the clinical condition of our models may be developed to represent healthy tissue or may include disease artifacts such as stenosis and calcification. Each model is fabricated to meet the needs of the specific project and may constitute one single continuous model or be of a modular nature to allow for adjustable positioning. To facilitate direct observation during testing, our models are fabricated from a translucent material and are fully compatible with our medical imaging modalities such as X-ray and ultrasound. Next slide, please, Mark. Equally important to understanding the clinical condition is the ability to accurately replicate the physiological conditions within the anatomical models. Through our research, we have established the know-how to develop and build advanced simulation systems capable of replicating physiological parameters such as pulsatile flow, heart rate, flow rates, and physiological pressure ranges. Hemodynamic conditions are reproduced through the use of a blood mimicking fluid, which exhibits the same density and viscosity as blood, and where required, particles may also be used to represent blood cells to allow for the ultrasound analysis of blood flow dynamics. Our simulation systems are designed to generate realistic flow waveforms, flow rates, and heart rates, accurately representing the physiological parameters of pulsatile flow, and can be programmed to include arrhythmias and fibrillation. Each system is closely monitored by instrumentation such as pressure and flow sensors to verify that the correct parameters are applied through the simulation system and model. Next slide, please. It's no secret that in vitro testing is central to the effective evaluation of testing of medical devices. An in-depth understanding of the anatomy, the clinical condition, and the use environment is vital for the design and development of any medical device. Conversely, the ability to subject prototype devices to challenge conditions in a consistent, reproducible manner is key to understanding their functionality and performance. And in vitro testing may be used throughout the device development cycle and beyond, from helping to inform the design process right through to the clinical demonstration of the product to potential customers. It can provide effective evaluation of prototypes as well as appropriate challenge conditions for design V&V. Requirements laid out by regulatory authorities mandate that medical device manufacturers demonstrate both the safety and efficacy of their devices. They also recognize the role that in vitro testing plays in device verification and validation, and commonly stipulate that devices be tested in vitro simulation systems that accurately mimic in vivo physiological and anatomical conditions. These test environments are typically representative of worst-case challenge conditions, and having the right clinically representative model is central to achieving this. The use of in vitro testing also comes with significant advantages, such as allowing for full customization of the testing environment, ensuring reproducible test conditions for device design iterations, and the convenience of having a clinically relevant test environment in-house. It also represents an efficient and cost-effective way to verify device performance up to, and in some cases, replacing testing in animal models. Next slide, please. Next slide, please, Mark. Yeah, and one, you can go 2 more there, please. Another one, yeah. Thank you. To demonstrate what can be achieved with in vitro testing, I would like to highlight a case study with one of our longest-standing industrial collaborators, Cerenovus. Cerenovus had developed an innovative clot retrieving device designed to treat patients with acute ischemic stroke. Prior to carrying out costly in vivo trials, the company was required to verify device performance through a series of in vitro studies. No cerebrovascular model was commercially available that simulated the specific anatomical challenges relevant to testing their device. The Met Gateway and Cerenovus collaborated on an Enterprise Ireland Innovation Partnership project to address this technology gap by developing a bespoke in vitro simulation system specifically designed for testing the performance of their product. The Gateway's research staff worked with the company to identify a set of performance criteria through medical image analysis of patient-specific cerebrovascular anatomy. Based on comprehensive review, analysis, and reconstruction of the clinical images, various model designs were selected and fabricated. Each of the models closely mimicked both the geometrical compliance and the frictional behavior properties of the native vasculature. A simulation system to incorporate the clinically relevant models was also designed and fabricated and was used to facilitate the in vitro evaluation of the company's device delivery and performances. Since completion of the innovation partnership, GMIT has maintained a high level of interaction with the company and has generated several patient-specific models with varying tortuous characteristics. Next slide, please, Mark. And again. Complementary to our capabilities in modelling and replication is our suite of medical imaging equipment. This includes a purpose-built fluoroscopy suite, ultrasound equipment, and an SEM microscope that can be used for histological analysis. Sorry, just back 1 slide, please, Mark. Thank you. Utilising our medical imaging equipment in parallel with our anatomical modelling and physiological replication capabilities allows for a clinically relevant simulated use environment to be established. Under these conditions, the intended use of the device may be replicated and its performance can be assessed in areas such as radiopacity, navigation, delivery, and accuracy of deployment. Next slide, please, Mark. One example of how MET has successfully replicated the clinical environment is through the development of a medical training model. A need was identified by one of our clinical partners to develop an accurate training model for administering regional anesthesia in the arm using ultrasound guidance. The models available at the time consisted mostly of homogeneous materials and were not truly representative of the anatomical structures in the arm. The team at GMIT were approached to develop a model that could incorporate all aspects of the anatomy to allow clinical personnel to be trained in the identification of the anatomical structures and to deliver anesthesia to the correct target. Developed by a team of researchers at Met, this phantom is designed to simulate muscle layers, bone, fascia, nerve tissue, and blood vessels in an anatomically correct arrangement, closely replicating that found in the clinical setting. The model was developed so that the shape and location of the anatomical structures vary along its length, aimed at representing various patient anatomies in one model. The model may also be used for training in vascular access. It allows for flow through the simulated vessels for use in Doppler examinations and to provide positive fluid flow when a vessel is successfully cannulated. One of the key challenges in the development of this model was material selection. Not only was it important to ensure the phantom accurately represented each structure as visualized under ultrasound, but we also needed to replicate the realistic motion and response of the muscle layers and connective tissue when compressed under the ultrasound probe during examination. Most importantly of all, it was critical that the phantom provides appropriate tactile feedback when advancing the needle to the target site, in particular as it passed through the muscle layers and through the fascia encapsulating the vessels and the nerve. This required careful balance of material properties, and the specific material recipes were developed for each element to ensure all requirements were addressed in the final model design. Animal tissue models were used to establish the forces required to advance the needle through each element, and the performance of the model was validated against this data. After successful development of the model, the technology was licensed to TruCorp, a supplier of medical training mannequins and simulators based in Northern Ireland. It is currently for sale as the TruNerve block training model. The team at Met worked closely with TruCorp during the transfer of the technology, aiding them in establishing their manufacturing processes and ensuring all elements of the design were incorporated into the final model. Next slide, please, Mark. And again, thank you. And again, please. Thank you. Met uses in vitro simulators to analyze device performance, and this clinical case study is an example of a study undertaken to review the clinical performance of medical device. The multi-layer flow modulator by Cardiatis is a flow diverter used in the treatment of aneurysms. Its braided structure transforms the recirculating blood flow in the aneurysm into laminar flow, thereby reducing the stress on the artery wall and reducing the risk of further dilation or rupture. It is constructed of a porous wire mesh, which ensures that that branch patency is maintained, thereby preserving downstream perfusion. A global registry on the use of the MFM in complex thoracoabdominal aortic pathologies was conducted, led by Professor Sharif Sultan of the Western Vascular Institute at University Hospital Galway. From a total patient cohort of 384, the MFM was used for the treatment of complex aortic arch aneurysms in 35 of the cases. Complex aortic arch aneurysms are extremely difficult to treat, often with less than favorable outcomes. Conventional treatment by open repair is associated with high morbidity and mortality rates. Hybrid repair is complex and largely limited to high volume centers and can be prone to end leaks, kinking and migration, while TEVAR stent grafts are commonly not suited for the aortic arch or areas with branching. The results from the registry suggested that in some cases, the MFM was not successful in stabilizing this particular pathology. This study was undertaken to better understand the performance of the MFM as a treatment therapy for these complex cases. Of the 35 cases in the registry, 6 of them were selected for analysis. 3 of the cases were deemed clinically successful and 3 were deemed clinically unsuccessful, and the selected cases constituted a mix of saccular and fusiform aneurysms. Next slide, please, Mark. The study began with understanding the preoperative clinical condition of the selected cases. Following analysis of the preoperative scans, patient-specific in vitro models of each case were fabricated from the data obtained. Each model was then placed in a flow simulation system and physiological flow conditions were applied. To create a meaningful comparison of preoperative and postoperative data, an analysis of the flow conditions in each model was conducted. This provided an understanding of the preoperative hemodynamic conditions and in doing so established a baseline for each case. This analysis confirmed the expected flow conditions within the aneurysms. The presence of characteristic recirculations within the aneurysm was successfully demonstrated, and the impact of the flow on the aneurysm wall was confirmed. Other physiological parameters obtained from the scans were also evaluated. A comparison of the center lines of all 6 cases demonstrated that there was no statistical difference between them. Next slide, please, Mark. Following the establishment of the preoperative condition, the postoperative scans for the selected cases were then evaluated, with a particular focus on the positioning of the device in each case. MFM devices were then placed in the patient-specific in vitro models so that they reflected the clinical scenario as accurately as possible. Each model was then placed back in the flow simulation system, and physiological flow conditions were again applied. The post-operative hemodynamic conditions for each case were evaluated and compared to the pre-operative baseline previously established. Next slide, please, Mark. The results of the study successfully demonstrated the clinical results seen in the registry, and the characteristics of both the successful and unsuccessful cases were accurately replicated. In the successful cases, the study demonstrated that the flow impact against the sac wall was eliminated by the MFM, leading to sac shrinkage or stabilization. For the unsuccessful cases, continued flow impact against the sac wall was confirmed, which would contribute to continued dilation. The position of the stent was shown to have a correlation to the results of the case, with areas of high stent porosity demonstrating less of an effect on the local flow conditions. Other device performance parameters were also evaluated, such as branch patency, which was confirmed to be preserved in all cases. Central to the success of this study was the development of accurate in vitro models and the establishment of a physiologically accurate flow simulation testbed, allowing for the successful analysis of the pre and post-operative flow conditions. Next slide, please, Mark. So that was a snapshot of what can be achieved with in vitro models. At MET, we are continuously striving to further our knowledge and capabilities in vitro modelling and testing. And so the inevitable question, where to next? Central to the development of the capabilities of MET is the launch of our next generation medical imaging suite. Not only would this provide state-of-the-art medical imaging to our industry, research and academic partners, but it will also allow us to expand the potential of our models and simulators. Our new Philips Azurion 7 M20 digital fluoroscope is currently under installation at GMIT's Galway campus. Representing a significant investment by Enterprise Ireland and kindly supported by GMIT, this constitutes an outstanding resource for the MedTech industry and academic and clinical researchers alike. Not only will this facility be used for device evaluation and testing, it will also be used to demonstrate and provide training on new technologies and devices for clinicians and staff. The system can also be used for training personnel in imaging technologies and the interpretation of medical images. It will include a conferencing facility for streaming live images to external parties, such as off-site engineering teams, physicians, and research partners. The Philips Azurion 7 M7 fluoroscope is optimized for vascular, non-vascular, cardiovascular, and neuro applications. With its high resolution and large field of view, it offers superb image quality to evaluate small details and vessels with clarity. The system has a 20-inch flat panel image detector, which can be rotated over 90 degrees to facilitate both portrait and landscape views. With Clarity IQ technology and zero dose positioning, it provides reduced dose exposure, enhancing operator and staff safety. The FlexVision XL HD 58-inch monitor offers exceptional viewing and can include displays from third-party equipment or other imaging modalities as desired. While parallel working also allows team members to view and analyze images in the control room without interrupting an ongoing test in the examination room. Expert CT Joule facilitates the acquisition of 2 scans at defined intervals, providing CT-like images, and can aid in the assessment of soft tissue, bone structures, and stent deployment. And I'll now take you through some of the other key features of the Philips Azurion 7 M20. And next slide, please, Mark. The Philips Azurion 7 is optimized for vascular applications and has several features for enhanced imaging. The 2D quantitative vascular analysis feature facilitates the quantitative assessment of aortic and peripheral vasculature. Based on 2D angiographic images, it can quantify vessel sizes between 5 and 50 millimeters in size. It allows for automated vessel segmentation and provides for the measurement of the diameter along the selected segment. It can also detect obstructions within the vasculature and can quantify relevant stenosis characteristics such as diameter, length, and percentage stenosis area. Vessel Navigator offers a complete live image guidance solution for endovascular procedures, providing an intuitive and continuous 3D roadmap to guide you through the vasculature. Using 3D roadmap, you can also create an overlay with a 3D contrast enhanced angiography volume. It provides real-time 3D views of device advancement through complex vessel structures. It also features automatic motion compensation for neuro exams. Smart perfusion helps assess the changes in perfusion due to an intervention, be that vessel patency after endovascular intervention or evaluating tumor perfusion after embolization. Next slide, please, Mark. And one more, please. Heart Navigator is specifically designed to support a wide variety of structural heart disease procedures. It provides automatic segmentation of anatomical structures, landmark points, and planes from previously acquired CT scans, and can automatically detect and segment calcifications within the anatomy. It includes functionalities such as automatic disease distance, diameter, area, and perimeter measurements for TAVI and TAVR procedures. It also provides segmentation, measurements, and viewing angles for other structural heart disease procedures, provides live image guidance through 3D overlay on the live X-ray image. Different visualization tools, including anatomical landmarks, viewing planes, and measurements, are available to support precise planning for mitral valve replacement and left atrial appendage closure procedures. It also includes virtual device templates to allow for accurate sizing and virtual target positioning before device deployment. And next slide, please, Mark. We are continuously expanding our capabilities and have successfully secured further funding from Enterprise Ireland to purchase compatible ultrasound equipment to be used in conjunction with the fluoroscope. This ultrasound will be used in a fully integrated manner through the use of the Echo Navigation software. Echo Navigator is a unique tool optimized for the structural heart disease interventions. These procedures often rely on X-ray imaging to visualize the devices, while also using transesophageal echo imaging to visualize soft tissues and anatomical structures. These images are typically acquired and displayed separately, requiring mental alignment by the user. Echo Navigator is a real-time imaging product that can enhance the procedure by combining both X-ray and 3D transesophageal echo in an interactive, intuitive, and procedurally relevant way. It is designed to help you guide your device in 3D space more quickly. It allows for multiple user-defined live views of echo data, showing several relevant anatomical structures from different angles simultaneously in real time. The image orientation of the C-arm view synchronizes the echo images with the X-ray images, and the echo viewpoint is automatically adjusted as the gantry is repositioned. Markers may also be placed on the soft tissue anatomical structures in the echo image, and these markers will automatically appear in the X-ray image to provide context and help guidance. The dual and overlay imaging capabilities of the Echo Navigation system allows for the accurate assessment of device deployment accuracy, while at the same time assessing, for example, their interaction with the device with the adjacent soft tissue. Next slide, please, Mark. Every project we undertake, be that applied or academic research, brings new challenges, and with it new opportunities for the development and expansion of our expertise and knowledge. Through our research, we continue to grow our understanding of the clinical condition, allowing us to continually improve our modelling and replication techniques. Our most current work includes improving the transparency of our models, addressing the frictional properties of our silicone models, and pushing ever further towards modelling smaller and more complex vasculature. We're working on improving the tactile feedback of our models to ensure they provide accurate representation of the clinical scenario. Our future work will include the development of hybrid models, including animal tissues where appropriate, to facilitate histological assessment and to gain a better understanding of tissue response. We are also looking at alternative ways to replicate the action of the musculature through the development of soft robotics and electroactive polymers. The output of our research is leading to the development of new therapies, and we will continue to inform clinical practice through our research output and publications. Next slide, please, Mark. And again, yeah. I would like to acknowledge the staff and students of the Centre and also our conferred postgraduates whose work has contributed greatly to our current offerings and also our clinical partners. Next slide, please. So thank you very much for your time. I would be happy to take any questions you might have.
Thanks, Sharon. So I have the dashboard of the questions here now. So the first one, do you have any experience using MRI or CT images to create accurate 3D computation models, for example, finite element analysis? And if you do, where do you source the MRI CT images? Do you have or use a database, or do clients supply the images specific to the application?
We would certainly have experience in working with MRI and CT scans, in particular in terms of obtaining anatomical or clinical data to generate our models. We have limited experience in CFD. It isn't a technology we typically offer. We would have done a certain amount of it in the past, but it wouldn't be among our core capabilities. In terms of sourcing the scans, it can vary between, if we are working with a particular industry partner, they may have a particular scan that they want us to replicate on their behalf. And we do have a repository, a certain repository of clinical data that we can pull from for certain models. Sorry, Mark, I can't hear you there.
I had myself muted, apologies. So thank you for the presentation. I'm curious as to what you adjust to improve tactile response.
At the moment, we're looking at how we actually house the models themselves. So a lot of the models that we have are just supported kind of at certain points along the length of the model. So we're looking at ways of housing that so that the tactile feedback would be improved. Sorry, Mark, I can't hear you there.
Right, yeah, sorry. So what are the key parameters that you need to assess when developing an anatomical model?
So for us, really, the biggest question is always understanding what it is that you want to replicate and what is the question that you want to ask of your device. What we find a lot is trying to replicate perhaps too much in one particular model, and you end up with a model that doesn't really address the question that you necessarily need. Developing a deep understanding of the clinical condition itself, and that's typically through the analysis of clinical data, and understanding what it is that you're going to use the model for.
Okay, so next one, sorry. Can you make up tortuous models if we give you a segmented CT file? What materials do you use, and are they just silicone, or is there any new materials that you have?
Yes, absolutely. We specialize in making patient-specific models, so we can certainly make a tortuous model. That's no problem. Our models are primarily silicone, and certainly in particular, the models that replicate the vasculature would be silicone-based. Yes.
To follow up to that, so the person is interested in issue with device kinks due to patent anatomy.
Okay, so this person is looking to, I presume it's a particularly tortuous anatomy that this person is looking to replicate. I mean, we can certainly help in terms of device assessment as well, in terms of what's causing it to kink and what particular anatomies may be causing difficulty with that particular device. If that person would like to reach out to us, we'd be very interested to kind of talk through the problems that you're seeing and see how we can help.
Okay, thank you. So a follow-up from somebody else. Have you experienced a patient with too much torsion, etc., in the vasculature that you could not model?
Not to my knowledge. Our limitations tend to be more in terms of the size of the vasculature. And again, that's something we're continually trying to improve. We can currently model down to about 1 millimeter in size, and we are looking at going beyond that. But I'm not aware of a tortuosity that we failed to replicate to date.
Okay, and then building on the tactile feel question, are you looking at the inner material of the model to reduce friction?
Yes, we are. That is something we get in terms of feedback from a lot of our clients in terms of friction, and I suppose it's well known that it's an issue that silicone models are plagued with. So yes, we are looking to address the friction of the inner lining.
And another question, we've observed challenges where anatomically correct models do not identify superiority of deliverable between prototypes. How do you approach differentiation versus anatomical correctness, I assume that's supposed to be?
Sorry, Mark, could you repeat that, please?
So we've observed challenges where anatomically correct models do not identify superiority of deliverability between prototypes. How do you approach differentiation versus anatomical correct? I assume that means correctness. Maybe I'll skip on, and the person who wrote that question might just put in some more detail to me, if that's okay, and we'll come back to it. Sure. So a simpler one, when will the fluoroscope be up and running?
So as I say, it's currently under installation, and as with everything, it's always the paperwork at the end that's kind of the longest part of it. We're hoping January at the latest. With a bit of luck, we might be able to open in December, but January at the latest.
You're waiting for the license, isn't it, from the EPA? That's really what
exactly.
it is.
The license for the EPA, and we've training lined up for the next couple of weeks with our people as well. So, but the biggest thing is the license, yeah.
Okay. You mentioned a study regarding clot trajectories. What experience does Met have in fabricating clot trajectories?
So we fabricate all our own clot analogues in-house, and they are mammalian-based clots, typically ovine, swine, and sometimes bovine. And we have done a suite of mechanical testing on our clot analogues to show the close correlation between animal and human clots. So we've quite a lot of experience in manufacturing clot analogues.
Okay. That appears to be the end of the questions that we have. So we'll hang on maybe just to see if any more come in, or if somebody wants to. Oh, sorry. So what is the typical endurance and test duration for the physical model, heart model more specifically?
I presume I'm taking that to be of the silicone models. So they are generally quite robust, the models. Of course, it will come down to the wall thickness. So if you're looking to model a particularly small vascular wall, then obviously the robustness of the models will be reduced. If you're looking specifically at a heart model, they're generally quite large and quite robust models, and we've yet to see a failure on any of them.
Okay, thank you for that. So look, that looks like it's, sorry, more coming in. Do you have experience in other autometries outside of CV?
Yes, we do. Just the example I gave there of the training model that we've done. So we have modeled the musculature from an ultrasound visibility perspective. We have done models of the urinary tract. We've done models, obviously we've done the neuro models as well. So yeah, we do. We operate outside of the vascular space. I suppose perhaps the presentation was a little misleading, that it suggested that that was really all we do, but no, we do operate absolutely outside that space as well.
And do you have experience in lung models?
We do. We had a project a couple of years ago where we actually used resected swine lung models, and we had a ventilator that we could actually replicate the movement of the lungs. It was actually a training model that we generated as part of a project with University Hospital Galway.
Okay, all right. So I think we've probably come to a good natural point to end that. If other questions do come in, we'll ask Sharon to answer them and to get back to people. So again, Sharon, thank you very much for the insightful presentation today, and thanks for everybody online for attending. Our next webinar will be in late January, the 28th, and it'll be focusing on augmented reality and virtual reality. So hopefully we'll see some of you there. And then, again, thanks very much. Just to point out that the webinar has been recorded and will be available later on. So with that, I'll say goodbye and thank you all.
Thanks very much.